Good morning. Today's class is on lab diagnosis of viruses. The previous class you had on an introductory class on viruses where you were told about the different types of viruses, the symmetry of viruses, etc. So today's class is on lab diagnosis of viruses. There are three general approaches for the diagnosis. You can have a direct detection of the viruses, or you can isolate the viruses, and lastly the serological tests. When it comes to the direct examination, the light microscopy plays an important role, wherein you are going to look for the histological appearances and the presence of inclusion bodies. The second modality is antigen detection, which can be done by immunofluorescence and ELISA. The third is when it is difficult for you to see it under the light microscope. To study the morphology of the virus particles, you can use the electron microscopy. The last examination, direct examination of the viruses, is by viral genome detection. Either you can use a specific nucleic acid probe or a PCR, polymerase chain reaction. So in the light microscope you saw inclusion bodies. This is an important topic for you all, where it can be asked as a 3 marker, which is quite commonly asked in the university exam. These inclusion bodies are nothing but crystalline aggregates of virions, or these are made up of the viral antigens at the site of virus synthesis. These are most often visible under the light microscope, and they are produced during the viral replication within the host cells, and they are quite big in size, therefore can be seen under the light microscope. These inclusion bodies can be classified based on where they are present. Either they can be intracytoplasmic inclusion bodies, intranuclear inclusion bodies are those inclusion bodies which are present both intracytoplasmic as well as intranuclear inclusions. The examples of intracytoplasmic inclusions are smallpox. The infections where you see these intracytoplasmic inclusions are smallpox, which are called as Guarneri bodies, rabies, Negri bodies, yellow fever, which are called as councilman bodies, foulpox, Bollinger bodies. You can also see them in diseases like molluscum contagiosum and the respiratory syncytial virus infections. The intranuclear inclusion bodies, these are divided into 2 types, Cowdrey A and Cowdrey B type of inclusion bodies. Cowdrey A type are variable, granular in appearance, and examples of viruses or viral infections which produce these intranuclear type Cowdrey A type inclusions are herpes virus, yellow fever virus. The Cowdrey B are much more circumscribed in nature, and the infections which produce these are adenoviruses, polioviruses, and cytomegaloviruses. The inclusion bodies which are present both within the intracytoplasmic and intranuclear, the example is measles virus. The second one is the immunological detection of viral antigen, wherein it can be done by using the fluorescent antibody, enzyme immunoassays, radioimmunoassays, the latex agglutination test, and all these detect the viral antigen in patient's specimen. This is just to show you the fluorescent antibody test, where you can do it both direct and indirect immunofluorescent methods can be done, where you know that direct is always an antigen detection and indirect is usually an antibody detection, where you have the nuclear and cytoplasmic staining patterns which are quite typical for influenza virus, adenovirus, and herpes virus. Viral serology, the primary means of diagnosis until the 1970s. Yes, it was used for detection of immune status, but now these serological tests play an important role in detecting the immune status. And when the virus, another indication is when the virus cannot be grown or detected by immunoassay or molecular assays, and usually we are detecting the IgM and IgG antibodies. IgM is always detectable in acute infections, and somewhere between 7 to 14 days the sample has to be collected for you to detect the presence of specific IgM antibodies. And usually we take it as a diagnostic titer whenever there is a 4-fold antibody titer between the acute phase sera versus the convalescent sera. So for an acute phase sera, you're collecting it immediately during the immediately after the onset of the symptoms, that is for the detection of IgM antibody. And for the convalescent sera, you're going to wait for about 2 to 3 weeks after the acute onset to collect the sera. So you should have a 4-fold raise between these two. So this is just to show you that the serological assays which we are doing is to measure either the binding antibody to the viral antigen, like you may have an enzyme immunoassay or radioimmunoassay, or you can also do a Western blot technique. The third method is when you're going to detect the viral particle as such. Sometimes when it is difficult for you to grow the viruses, this is one of the indications where you can detect the shape of the viral particle. In the previous class, during the introductory class, you were told about there are or given examples of viruses which have specific shape. For example, if you want to see that you can have, if it is a rabies virus, you have it in a bullet shaped. If it is pox, you have it in a brick shaped. Ebola, filamentous, and rotavirus, a spoke wheel type. Astroviruses, star-like viruses, examples are these. So usually these are effectively used effectively in detection of viral agents of gastroenteritis which cannot be cultivated by cell cultures. So examples of viruses which you can see in these slides are rota, adeno, corona, and astroviruses, just to name a few. The last method is the molecular detection, where example you can do it for papilloma virus or hantavirus, HIV virus, etc. And you can either buy the probes and then do it, or you can have an in-house molecular assays, wherein the generation time or for you to give the report is around 2 to 3 hours. Next is the virus isolation. As we all know, viruses are obligate parasites, so therefore they need a living cell for their replication. Unlike bacteria, where you are using an artificial media for cultivation of bacteria, in viruses you cannot have that, so therefore there are various modalities by which you can isolate the viruses. One is the animal inoculation, use of embryonated egg, and thirdly, which is most commonly used, is the cell cultures. So in animal inoculation, we are going to look for a visible lesion which the virus may produce, or the disease which it produces, or lastly, it may lead to death of the animal. In embryonated egg, there are various parts of the egg which can be used, and for just to name a few, what are the changes which can occur in the embryonated egg? Formation of pox on the chorioallantoic membrane. For example, the herpes viruses, the variola viruses, all these can produce pox on the chorioallantoic membrane, and the differences between the formation of these pox can be used for identification of a particular virus. Or you can perform a heme agglutination test once you have harvested the egg. The last one is the cell culture, wherein you are using the tissues, which I will tell you all in detail how the cell cultures are done, and for you to detect the presence of the virus in a cell line, there are various methods. One of it is the cytopathic effect, the second is metabolic inhibition, heme adsorption, interference, transformation, and immunofluorescence. So these are the various methods by which you can detect the presence of a virus in the cell line cultures. So as I already told, for cultivation, they're all obligate intracellular parasites, hence cannot be grown on an artificial media, and they require the living cells for cultivation. Just to give you a brief historical events, in 1900 it was Reed and colleagues who used human volunteers for their pioneering work on yellow fever. 1930 Gay introduced the cell cultures. 1931 Goodpasture and Burnett used the embryonated hens for the first egg for the first time. So why do you need to cultivate the viruses? One is to isolate and identify what is the virus which causes the disease, to develop vaccines, to look for the pathogenesis, genetic and effects on the cells, for example, the oncogenesis, for research purposes. So these are the three various methods. You have animal inoculation. The animals which can be used are the mice, rats, monkeys, chimpanzees, ferrets, and human volunteers. For embryonated egg, there are various parts of the embryonated egg, or the duck egg, which is larger in size, which can also be used. Most often, duck's egg can be used for growing the viruses for vaccine production. The various parts of the egg which can be used are the yolk sac, allantoic cavity, amniotic cavity, and the chorioallantoic membrane. Tissue cultures are organ cultures, explant cultures, and cell line cultures, which are further classified, which the differences I will be telling you all, into primary cell cultures, diploid cell strains, and continuous cell lines. So this is just to show you animal inoculation, intraperitoneal inoculation into the mice. So these mouse, most often the mice which are used are the suckling mice, which can be used for growing the Coxsackie A and Coxsackie B virus, and the presence of these viruses can be identified in these mice by noting down what kind of paralysis these Coxsackie A and B produce in the mice. So if you see Coxsackie A, it produces a flaccid paralysis in the suckling mice, whereas B produces a spastic paralysis. The other viruses where you can use the suckling mice is the arboviruses and toga viruses. And you can inoculate the mice either subcutaneously, intraperitoneally, or intracerebral routes. Rabbits can be used for smallpox, herpes viruses, and rubella viruses. Hamsters for herpes, which produces a paralysis of both the hind limbs, and SV40 virus, which is known to produce subcutaneous tumors. Ferrets can be used for influenza virus, hepatitis A, coronavirus, and rhinoviruses. Monkeys can be used for polioviruses, hepatitis A, hepatitis B, yellow fever, and rhinoviruses. The disadvantages of using the animal inoculation: one is expensive and difficult to for the maintenance of the animals, and second important thing is most of these viruses are known to have latency in these animals. So whenever they have the latency, they are capable of producing disease for the most commonly the animal handlers or people who work in the lab along with these animals. They may get the infection. So therefore the latency plays an important role, and they can that is why the animal inoculation is not done. But and also that these viruses can be shed in various ways from the animals, say for example urine, feces, saliva, etc., and they can produce disease in humans. So therefore animal inoculation is not much used, probably except for the suckling mice for Coxsackie viruses. Coming to the embryonated egg, you can either use a chick or a duck embryonated egg. It was first used by Goodpasture and further developed by Burnett, and this picture shows you the various parts of the embryonated egg, and to identify these and to find out if there's any defect in the embryonated egg, we use a technique which is called as candling of the egg, which you can see in the picture lower on the slide. So when you put the egg in usually we make it with a say for example a chalk-piece box which can be covered with a black paper, a light put inside this box and a small hole in the center wherein you can place this egg, and when the light passes through the egg you can make out the various parts. So these are the various routes wherein you are going to inoculate an embryonated egg, and which also gives you, this picture also gives you what are the different types of viruses which have to be inoculated into the various parts of the embryonated egg. This picture is very important for you all, wherein when a 3 marker is asked, if you could draw this diagram, label it and write the viruses which can be isolated from these routes of inoculation, it will fetch you marks. So how do you detect the presence of the viruses or the viral growth in the various parts? For example, you have pock formation or chorioallantoic membrane, or you can have the death of the embryo, or you can also detect the heme agglutinating property of embryonic fluids. So what do you which viruses are inoculated into allantoic cavity? It's usually used for influenza vaccine production, yellow fever vaccine 17D strain, rabies vaccine Flurry strain, Newcastle disease, mumps and other paramyxoviruses, and avian adenoviruses can be inoculated into the allantoic cavity. Amniotic cavity is commonly used for primary isolation of influenza viruses and for mumps viruses. The chorioallantoic membrane, as I told you all initially, they produce pock, and the differences in the pock can be used for identification of the virus. As this table shows you, variola pocks are small, shiny, white, convex, non-necrotic and non-hemorrhagic, whereas the vaccinia ones are large, irregular, grey necrotic and hemorrhagic, whereas herpes, they are small, oval and non-necrotic pock which are produced on the chorioallantoic membrane. The yolk sac can also be used for isolation of mumps, Eastern encephalitis or any encephalitis viruses, influenza type A and B and herpes simplex virus. Now coming to the tissue cultures, these are from whole or part of the organs. The cells are allowed to differentiate and preservation of the architecture or the function of the cells. They are mainly employed to study the pathogenesis, effect of antiviral agents, and may not be used for a routine diagnostic purpose, but they are helpful for isolation of viruses in latent state. So what are the different types of tissue cultures? There are organ cultures, explant cultures and cell cultures. Organ are the small bits of organs are maintained in vitro for days or weeks, and these cultured tissue pieces, the architecture of the tissue is retained and the cells maintain the differentiated state. Isolation of the viruses which appear to be highly specialized to that particular organ can be isolated. To give an example, the ferrets tracheas are used to isolate the coronaviruses. Explant cultures are the disadvantage of using the organ was there was lot of contamination, bacterial contamination of these, and whenever there is bacterial contamination, it is difficult for us to grow the viruses. So the next they used explant cultures, wherein fragments of minced tissue could be grown as explants and they were embedded in the plasma clots. But they're not used as of now. To give you an example where explant cultures were being used is adenovirus for adenoid tissue for isolation of adenoviruses. The cell cultures are the most commonly used ones. These use These are defined as cells which grow in vitro under conditions in which the cells are no longer organized into tissues. They get dissociated into component cells by either using proteolytic enzymes like trypsin or collagenase and or through mechanical shaking. These cells are then washed, counted and suspended into a growth medium. So this method is used primarily for diagnosis of most viral infections. So these culture medium which is used is of two types, either the growth media or maintenance media, and which contains the growth media contains 10% serum, or maintenance media contains about 2% serum. Growth media is used for initiating growth of the cell, whereas the maintenance for maintenance of the metabolism of the cell. These cells which are dissociated and counted and poured into the bottles are kept moist and continuously supplied with nutrients. They are routinely incubated in roller drums and holder tubes tilted at 5 to 7 degrees and kept at 37 to 37 degrees Celsius. They are also supplemented with bicarbonates which act as buffer systems, and they have for us to detect, as I told you all in the earlier slide, there are various methods of detecting the growth of the viruses within these cell lines. So one of it is the metabolic inhibition, which we need to know by different change in the color. So therefore phenol red is used as an indicator. Antibiotics are incorporated so that they do not get contaminated. These are incubated for 1 to 4 weeks and periodically inspected under microscope for presence of viruses. So this is just to show you the picture where the tissue is taken, the cells are dissociated, they are counted, poured, and maintenance of them through roller drugs for aeration and use for introducing the specimen. So 3 different types of cell cultures are present. They are primary, diploid, and continuous cell lines. The primary cell lines are usually freshly taken from the body and cultured. These are capable of only limited growths, obtained by dissociation of cells from minced organs, and 75% of the cells have normal complement of the chromosomes. And examples of these are monkey kidney cells, rhesus monkey kidney cell lines, and human embryonic kidney cell lines. The diploid cell lines or semi-continuous cell lines, these are obtained with successful subculture of the primary cells monolayers. These consist most often by spindle-shaped fibroblastoid cells. They undergo the doubling time of about 50 to 100 populations, or generation of 50 to 100, and before they die. The cells of the single type that retain original diploid chromosome numbers. The examples are the MRC5 and W138 cell lines. Continuous cell lines, these are usually a single type of cell lines, and all these cell lines are derived by cancer cells. They are capable of continuous cultivation, and chromosome complement is heteroploid cell lines. Examples of these are HeLa cell lines. This is probably one of the oldest continuous cell lines which are derived from a patient by name Henrietta Lacks, who had cervical cancer and who died in 1951. So the advantages of cell lines: the number of cells required to initiate cell culture is very low, the requirement of serum or nutrient are less, growth rate is faster, and contact inhibition is lost. The disadvantage is the cells tend to overgrow, require more frequent attention. So this slide, just to show you all the difference between the primary, diploid, and continuous cell lines. As I told you all the characteristics, the number of passages which can be done, less than 10 for primary, 50 to 100 for diploid, and continuous for the cancer cells which are used for continuous cell lines. And the second column shows you the various examples of these different cell lines and where they can be used. This is another method of shell vial culture, the modification of a conventional cell culture. This is wherein you are going to use a fluorescent labeled antisera, that is which contains the antibodies which can be used for confirmation. And why we use this when compared to the conventional technique is this is more rapid when compared to the conventional cultures. So the various methods for detection of viral growth in cell cultures, you have cytopathic effect, metabolic inhibition, heme adsorption, interference, transformation, and immunofluorescence. So cytopathic effect is, there is a change in the cell line which you have used. That is, you're comparing with an uninoculated monolayer cell line versus the one which have inoculated with a sample containing a suspected virus. So to give you a few examples of the cytopathic effect which some viruses have. For example, you have one of the features is there is cell rounding, degeneration, aggregation, or loss of attachment of substrate. So to give you an examples of these, if you see the enteroviruses, if you have a sample which contains enteroviruses and you inoculate it into the monolayer of cell lines, you will see that the entire cell sheet gets degenerated. Whereas your herpes virus will have discrete focal degeneration. Adenoviruses produces clumping of cells which resemble a bunch of grapes. You may have some viruses which produces syncytial formation, which is usually seen in measles virus. Just to show you some of the examples where there is rounding of cells, aggregation of cells, and focal degeneration of cells. Now coming to the metabolic inhibition, normal cell culture medium turns acid due to the cellular metabolism. So when a virus is, a sample containing a virus is introduced into the cell line culture, if the virus is growing, the cell metabolism gets inhibited. So the no acid is produced. So this can be made out by change of the indicator which you have incorporated into the medium. The other property is heme adsorption. Some of the viruses have heme agglutinins on the surface of their viruses, and these will produce, if you add RBCs to them, they will produce the aggregation of RBCs. This property is called as heme adsorption, and some of the examples which produce this heme adsorption is influenza viruses and measles viruses. Interference. So this is nothing but growth of a non-cytopathogenic virus in cell culture, which can be tested by subsequent challenge with a known of cytopathogenic virus. What will happen now? The growth of the first virus will interfere with the growth of the second virus. So as we know, the second virus, what are we going to introduce? A virus which we know that it is going to produce a cytopathic effect. So when such a virus is introduced into a cell line, if the cell line already contains a virus which is multiplying there, you will not see any cytopathic effect. Okay, so this is called as interference. The fifth one is transformation. Oncogenic viruses. These induce cell transformation. So whenever there is a cell gets transformed, there is a loss of contact inhibition. Because of this loss of contact inhibition, the cells will appear growing on one top of the other, and they form what we call as microtumors. So the last method by which we can detect the presence of virus in a cell line culture is immunofluorescence. As I've already explained to you all the shell wall culture, this is just a small modification where cells from the viruses with the infected cultures are treated with a fluorescent conjugated antisera. Antisera means it contains the antibodies and examined under the microscope, and they will fluorescene if it is a homogeneous antibody. Okay. So these are the various methods by which we can detect the presence of viruses in a sample.